discussion board, so I would imagine that everything was completely 100% clear from the last lecture, right? Okay, yeah, okay, so you put stuff up there, though, because this keeps me amused answering them. One question, actually, that came up during the lecture that I sort of posed for myself was that I told you that the cell body of neurons is the site of protein synthesis, because that's where the nucleus is, and the endoplasmic reticulum, for the most part, is rough endoplasmic reticulum. And that every year I do this course, once I've said that, I sort of wonder whether there's been any progress as to whether protein synthesis can also occur out in the dendrites of neurons. And when we talk about learning and memory and about changes in the strength, sort of induced changes in the strength of synaptic connections, you'll see why the possibility that neurons can actually synthesize proteins out there from messenger RNA and control that synthesis might be really interesting and important. So it turns out that yes, there has recently been some progress, and it's becoming better established that mammalian neurons, as well as insect neurons, have the ability, to some extent, in some cases, to synthesize proteins out in their dendrites. And that synthesis may depend upon the incoming activity of the synapses that impinge on those dendrites. Okay, so make a note of that, because that's something that I think is going to be quite interesting and important for the future. And hopefully when we talk about learning and memory, even if it's not in the book yet, we can talk about, you'll see immediately, I think, why that might be important. Okay? All right, so we're still going through neuronal cell biology, and for the next few slides, I really want to just emphasize that all of the techniques that you've come across in terms of being able to label cells with specific kinds of dyes and determine what proteins are expressed and what are not, all those general techniques can and have been applied to neurons. And in terms of neuronal cell structure or ultrastructure, one of the main take-home messages is that neurons, as you can imagine, are highly polarized. In other words, what the proteins that are expressed, or the proteins that are present in the dendrites and are often different from the proteins that are in the axon, obviously. And again, the axon and the dendrites may contain special proteins that are not at all present in the cell body. So let's go through some of these techniques and quickly and look at the results that can be obtained, specifically with neurons. The structure of neurons can be examined by immunostaining, that is the use of tagged antibodies, fluorescent antibodies, for instance, that are targeted against specific proteins, and by what's called GFP tagging methods. Anybody know what GFP tagging is? Yeah, go over that. I'm waving. Was that just a reflex jerk?
I think it sort of works, but green fluorescent protein, and it's like using sort of like immunostaining, where there's like GFP is like bound to another antibody that binds to protein of interest.
No. Good try. So he says that you're right about the GFP. The green GFP stands for green fluorescent protein, and what you did say is protein from jellyfish. Actually, it's a jellyfish protein. It's what makes jellyfish glow in the dark in the sea, just after sunlight. But the question is, it really doesn't have much to do with immunostaining. And the thing you didn't address is how do you get the GFP to quote unquote bind to the protein of interest, yeah? And that's really the crux of the issue. So does anybody want to address those two? Yeah.
You take the gene for green fluorescent protein and you insert it very close by, so the gene that codes for whatever protein you're trying to examine. So they're very close by to each other, and then it's almost like one cohesive protein that's created.
So you take the gene for the green fluorescent protein and you insert it. I guess that involves some genetic engineering. You insert it into the genome of the target animal, close to the protein that you're interested in finding out where it's expressed. And what the cell expresses then is a combination, is actually called a chimera. It's the protein that you're interested in with the code for the structure of the GFP attached physically, just sort of linked to it in the polypeptide chain. And so the thing fluoresces, right? Not quite. No, it would be good for some. There's just a methodological issue. I had just a methodological issue with what you said. Concerns, again, how do you get the GFP joined to the protein? And I agree that what you said would work, but it's not actually always or often the way it's done. So yeah, what's the catch?
Can't you attach the GFP gene to the promoter directly?
That's a different technique. That tells you, because can't you just drive the GFP gene with the promoter for the protein of interest, right? In that case, the cell will glow sort of generically everywhere when the protein is expressed, when the promoter is activated. So that's rather an indirect way of telling whether the protein is being expressed inside the cell, period. But it doesn't tell you where the protein is expressed, which is the question we sort of posited about neuronal structure, right? Because what it tells you is that the messenger RNA is being produced if you just attach it to the promoter. So you do have to attach the GFP physically to the coding region of the protein, as you said. But the question is, how's that done? What's to do with this business of inserting it? What do you insert into the genome of the animal that you're going to do? Do you think it's easy to find where a gene is expressed in an animal and then target the GFP to a particular point in that gene? Is that a real easy thing to do? Yeah.
Do you have to use homologous recombination?
You would have to use homologous recombination for that. You'd have to recombine, but it's actually not trivial for every lab to do that. And if you don't have a genome for the animal, it's even more difficult. Yeah.
Use a plasmid.
Exactly. So what you do is you obtain the messenger RNA for the protein of interest, and you insert a DNA copy of that into a plasmid, a bacterial plasmid, and then you do your stitching of the GFP gene to the protein, the coding for the protein of interest, and then you insert a plasmid with that chimeric coding in it. You insert that into your target animal and drive its expression with a suitable promoter. Right, so the promoter that you're using may not even be the natural one, but that's what you might do. So, I mean, these are just different ways of going about the same business. Yes.
Can you explain the process again?
Yeah. So instead of adding the GFP to the end of sort of the natural gene inside the cell, you can actually cut out the natural gene and replace it with a gene that is the GFP joined to the protein of interest, and then drive the expression of that chimeric protein. Yeah? And so doing it that way is a little bit more common than trying to insert the GFP into the native gene. Yeah, you basically get rid of the native gene and replace it with an artificial construct. Yeah, so. So all of that's very technical, though. It's good to get a conversation going. All right. So an immunostaining is pretty obvious. You just make an antibody in a mouse or a rabbit that recognizes the protein of interest. You then use a secondary antibody that's fluorescent, usually, to bind to the primary antibody, and that allows you to detect, using fluorescence microscopy, where these things are. These methods localize proteins within cells. They don't just tell you that it's expressed, They tell you where it's expressed. And so the localization of quite a few proteins inside neurons turns out to be highly specialized. And even common cytoskeletal proteins are distributed differently in axons, dendrites, and at synapses. So even things like actin and sometimes even microtubule components are localized within cells. Right? So these specialized arrangements of cytoskeletal proteins, for instance, create a scaffold that then in turn localizes ion channels and proteins that bind or release neurotransmitters. So one of the problems for a cell like this that you can imagine is how do you keep the neurotransmitter receptor proteins up here in the dendrites, and how do you keep the proteins that create and release neurotransmitter vesicles down in the axons. And one of the ways that's done is that the whole cytoskeletal components of these two parts of the cell are different. And so different proteins attach to them, right? Bind. So here's an example here. This is the figure A is a typical neuron, and this is the neuronal cell body, and you can't see the axon because it dives out of the plane of the, maybe very faintly you can see it, it sort of dives out of the plane of the microscopic section here. And then here it is, here's the axon going down here. And this is a typical neuron stained with antibodies against tubulin, and tubulin is the primary monomeric protein that makes up microtubules. And the microtubule binding protein called tau. So tau is a protein that binds to microtubules, part of the sort of construction of this cytoskeleton, right, in neurons. And you can see in this case that tau, the red glowing protein, which is labeled in immunostained, I think in this case, by an antibody that fluoresces red, tau is restricted to the axon of the neuron and doesn't really appear in the neuronal cell body at all. So highly differentiated and polarized, and neither is tau present, notice, in the dendrites, even though there are plenty of microtubules in the dendrites. This tau protein only seems to attach to the microtubules in the axons. So the point of this is not to sort of go into microtubule construction or why this should be, just to show you that even a protein as simple as one that simply binds to microtubules and and microtubules are present everywhere, more or less, only seems to bind in the axon in this case. Very highly differentiated. Okay, so the second slide is the localization of actin. So actin is the protein that is responsible for being the building block of microfilaments, the other cytoskeleton protein here. And you can see that in this neuron here, actin is only really present in high concentrations at these little tippy bits on the end of the growing axons, and I think these are dendrites. They might be also axons, but I think they're dendrites. And again, the green stain here is microtubules. And so again, this very common cytoskeletal protein that's present sort of ubiquitously in other neurons is not present, is only present at the growing tips of these neurons in this suspension of cells here. Whereas actin distribution in non-neuronal cell lines, and there are a couple of examples here, is sort of radially symmetric. All right, it's not sort of expressed just in one little localized patch of the cell or just in the extensions of the cell of these two non-neuronal cell types. And sometimes the actin expression is even more localized. So this shows you a neuron which is being cultured. And I think these are the dendrites. This cultured neuron is sprouting dendrites in all directions. And if you look very carefully at the ends of the dendrites and use again an antibody against actin, the filament protein actin, you can see that actin is present, and therefore presumably microfilaments are present, in these little spines, these little bumpy spines that stick out from the dendrite there. So it can be even more localized than not just localized to the dendrite or to the axon or to part of it. It can be localized to a very, very tiny area. Okay, and again, I think the tubulin, which is the protein for microtubules, is pretty uniformly distributed in these cells here, and in a non-neuronal cell, again, comparison with a non-neuronal cell. And these little bumpy outgrowths of the dendrites are called dendritic spines, and we'll see they're very important in learning and memory in the brain. And then lastly, as you can imagine, at the tips of the axons, there are these very specialized synaptic contacts with the target cells. And in this case, what we're looking at, the black surface here, is the surface of a muscle cell. And we're looking at the axons, the branching axons, from two motor neurons that are contacting and driving the contraction of those muscle cells. And again, the green stain is something like tubulin, just a ubiquitous stain. The red in this case is a protein that's not present on the neuron, is actually present on the muscle cell underneath. And this is the acetylcholine receptor protein. It's a neurotransmitter receptor protein. So this shows you how when a neuron makes synaptic contact with its target cell, it alters the expression of proteins on the target cell as well, and localizes that expression pattern so that the target proteins, the neurotransmitter receptors, are localized directly under the synaptic contacts of the neuron. Okay, so this extreme specialization of protein targeting is sort of transmitted from neurons to their target cells, to other non-neuronal cell types. Okay, so those are just some pictures and general principles. Okay, now we're going to turn the page and talk about glial cells for a few moments. And there are a couple of slides here which I altered this morning as I was going through things in more depth, so watch out for those. They'll be released after the lecture. Glial cells are the other cell that makes up the nervous system. It's neurons and glia. But that having been said, there's a variety of glial cell types with specific functions. So glial cells in general are primarily thought of as support cells. And if I was giving this lecture 10, 15 years ago, I would probably say that that's what they are. They're just support cells. So what do I mean by that? They regulate the environment around nerve cells. There are 10 to 50 times more glial cells than neurons in your brain. All right, so in 1 in 50 cells in the average part of your brain is a neuron. All the rest are glial cells. And of course, it's the glial cells that generate cancers. General cancers are gliomas. They're not caused by sustained division, uncontrolled division of neuronal cells. They're caused by uncontrolled division of glial cells. All right? So they regulate the environment around nerve cells because they wrap themselves and insert their processes everywhere. They take up neurotransmitters sometimes, released from axon terminals. They play a role in defending the brain, some types of glial cells, and the spinal cord against injury and infection. All right, and this has a sort of a perverse corollary to it that sometimes glial cells actually impede recovery. So the fact that we have 50 times as many glial cells around the neurons and they're busy regulating the environment makes it very difficult to go in at the cellular level and, for instance, encourage neurons to grow, because those neurons, as they grow, will encounter glial cell barriers and things. So if your spinal cord is cut, you not only have to solve the problem of how do you make the nerve cells sprout new axons or whatever and regrow, you also have the problem of how do you persuade the glial cells to aid that process rather than inhibit it. All right, so that happens. And very importantly, glial cells are absolutely crucial in directing neuronal growth. And so if you want to understand and control nerve regeneration to some extent, you've also got to understand and control the glial cells. And so because of this and because of the link with cancer, glial cell physiology and cell biology have been extremely intensively studied. So glial cells can also, or some of the types, can be stem cells. In other words, they can proliferate and differentiate into new glial cells, and in some places they can differentiate into new neurons. There are a very small number of localized places in your brain, spots in your brain, where new neurons can be created from glial cells. Remember I said in the last lecture that the neurons themselves in mature parts of your brain cannot divide to create new neurons. But it turns out there are one or two specialized regions where the glial cells can act as stem cells and generate new neurons. And obviously this is of intense interest because if we can understand and control this process, we can get access to cells that, stem cells that can be used for things like Parkinson's disease and so on without having to go to embryonic tissue. Yeah.
When you say regulate the environment around nerve cells, what environment are they regulating?
I think we'll come across this a bit later, but well, firstly, the levels of neurotransmitters around neurons is an important part of their environment, but the ion concentrations. So they're very busy regulating the sodium and potassium concentrations around neurons, and I think I'll come to that in a moment. We can have a little sidebar on that. Yeah, but obviously that's very important, and the calcium concentration. Any other questions? So what do they do in addition to all of this? Do they actually transmit information in the nervous system? And actually part of the information carrying capacity of the nervous system, processing capacity of the nervous system. And the old dogma is that they don't carry information. What carries information are the electrical potentials inside nerve cells. But that is changing, and it's blurred. And I'll talk a little bit about how it's become blurred. But I still think it's probably almost certain that glial cells don't carry information over large distances within the brain. What I mean by large, I mean several millimeters, perhaps, or centimeters. No, several millimeters. Okay? But that's just my personal sort of opinion on this matter. So you have to realize as you go forward through neuroscience that new sort of roles for glial cells in the actual processing of information in the brain may be found, and also in new roles for in neurodegenerative diseases and other things. Okay. I mean, it's certainly true that glia are vital for normal information processing, because, for instance, by taking up neurotransmitters, they basically prevent signaling from continuing in a part of the brain forever. All right? They destroy the neurotransmitters that are released from synapses and shut down transmission in the long term. Okay, so you can see how blocking the process of taking up neurotransmitters by glial cells could have dramatic effects on the gross level of neurotransmitters in parts of the brain. And that's certainly the case, and a lot of psychoactive drugs target the transport proteins that glial cells use to move neurotransmitters out of the area around neurons and into their cytoplasm, the transmembrane proteins, right? But recently, astrocytes in particular, right, this is a subset of glial cells, have come to be recognized as not only support cells for neurons, but also as partners in fundamental neuronal processes. Yes?
So, what do they do with the neurotransmitters that they take up?
They will metabolize them.
They don't release them again?
They don't. It's difficult because this whole business of glial cells being able to release neurotransmitters is a relatively new subject. But they don't release them again. No. A lot of neurotransmitters that they take up are simply amino acids that inside the cell are present at high concentration and just are building blocks for proteins. Right? But outside the cell are relatively rare. And so neurons can secrete neurotransmitters, can secrete just amino acids and use them as neurotransmitters because they're rare outside of cells. Yeah? ATP is a neurotransmitter, for instance. Okay? Common inside cells, not common outside. And hence, neurons can secrete it. And so, glial cells would take up ATP, right? No problem at all. Or glycine, or glutamate, or many other sort of simple neurotransmitters. It's not true of all neurotransmitters. Acetylcholine is destroyed by enzymes in the synaptic cleft. We'll see. Okay. So here are some glial cells. They're really pretty, and like neurons, they're rather diverse. This is called an astrocyte for obvious reasons, starburst sort of effect. These are all membrane processes, right? Here's something called an oligodendrocyte. We'll meet that in a moment. And here's what's called a microglial cell. This guy is motile. It's capable of crawling along. And it has a much more limited sort of set of processes, microglia. And then there are glial stem cells, like this one here, which are capable of generating new neurons or capable of generating new glial cells, like these oligodendrocytes. Right? So those are glial stem cells. Right? So those are several classes. Let's start with the microglial cells. They're found in the cerebrospinal fluid and in the CNS. They're small, motile, phagocytic cells. All right. So what's a phagocytic cell? It's a cell that eats other cells, it engulfs other cells, all right, like an amoeboid kind of beast, and like macrophages, which you might have come across, right, in the rest of your body. It can engulf particles following injury. It can secrete signaling molecules that modulate the inflammatory response to injury, like cytokines. So it's to some extent part of the sort of immune system of the nervous system, the equivalent thereof. And these are mobilized and activated certainly after damage to the nervous system. And then as those microglia, the macroglial cells, the most prominent member of the macroglial cell sort of type or family are the astrocytes. And this shows you some astrocytes, and they're colored green here. And behind them you can see neurons, the neuronal cell bodies here. So you can get some idea of the relative density of neurons and glial cells. There's 2 glial cells and maybe 20 or so neuronal cell bodies visible there. But you can also see how the glial cells extend processes that wrap around and betwixt and between all of the neuronal cell bodies, dendrites and things in this picture. All right? So they're involved in potassium uptake around active neurons. Why is it important to do that? You know, too many carrots, vegetables, potassium uptake, yeah. Right, because potassium ions are responsible for the resting, or the level of potassium ions, the concentration in the outside versus the inside of neurons determines their resting membrane potential. Yeah? It has to be kept low. What happens if the membrane potential goes up in a neuron? Yeah. Fires a signal. Yeah. What kind of signal? What's the form of the signal? No, that's a bit too general. What's the signal called that gets fired? Yeah, it fires action potentials, right? Okay, so when the membrane potential, resting membrane potential rises above a threshold, an action potential is generated. Right, you all know that from either 207 or 330, okay? So it's important that the glial cells keep the level of potassium low outside neurons, or normal, so that you don't get lots of spontaneous activity. But why is it important that they do this when the neurons around them are active? What's that got to do with it? Yeah.
Is it because when the neuron fires its action potential, the potential decreases, so potassium flows out. So in order for the cell to regain its original threshold, you need to lower the potassium outside, I guess?
Okay, sort of mixture there. Yes, during the action potentials, sodium ions rush in, remember? And potassium ions rush out. All right? And that's actually what restores the membrane potential in the neuron. Right? Is the potassium ions leaving. Right? In the short term. But in the long term, if a lot of neurons are active in the same place in the brain, potassium will begin to accumulate outside the cell. And that will result in the effect that was described over there, whereby the membrane potential of the neurons begins to get affected. It begins to rise, become more positive, and then you get spontaneous activity. Okay? So, somebody asked, so what these glial cells are doing are sequestering the potassium that comes out of the neurons during the action potential. Okay? They're moving it from the extracellular space into their cytoplasm. And then when the neurons are no longer active, it will be recycled back to the neurons. What will happen is the neurons, well, how do the neurons get their potassium back in the long term? How do we do that? Sodium potassium pump, yeah. So in the long term, once the activity is over, the sodium potassium pumps are also chugging away, and they lower the potassium back from in extracellular space, and then it passively comes back out of the glia and goes through the pumps back into the neurons. All right, so it's a nice little cycle of activity. So these function as a sort of a buffer for the potassium ions. Yeah, what a buffer is, right? Like a sort of active kind of cellular buffer, like a pH buffer, except it's a potassium ion buffer. Yeah. Okay, they're not motile. They're just stuck there. The neurotransmitter removal around active synapses is done by proteins in these processes that move neurotransmitters across the membrane into the cell. They have a role in guiding axons to their target cells during development. Very often during development of the nervous system, a glial scaffold is first created, and then neurons move through the glial scaffold, migrate through the glial cells, because neurons can do that when they're very young. They can move themselves and their processes along and migrate along to where they're finally going to live in the brain. And the other big thing about macroglial cells, the astrocytes, is they're a substantial component of what's called the blood-brain barrier. And this is the barrier of glial cells, basically, and some endothelial cells in blood, that separates blood from the fluid inside the brain. And this is enormously important, this barrier, economically important. Why is it economically important? Yeah?
Because a lot of drugs can't get through the barrier, so they're essentially useless.
Well, a lot of drugs can't get through the barrier, and so not essentially useless. Nowadays, you can get around it, but they used to be useless. But more particularly, they can't get through the barrier, so you can't just ingest the drug through your stomach or inject it. You'd have to find some way of getting it into the cerebrospinal fluid or something like that. But also, actually, it's actually a plus in some cases, because it means that drugs that, for instance, regulate your heart by altering the response of the heart muscle cells to neurotransmitters don't significantly affect your mood by affecting neurotransmitter receptors in the brain. Yeah? So some of the side effects of common drugs that are acting on our body, general body, are because those drugs have some ability to get through the blood-brain barrier and also and are to some extent psychoactive. Yeah? Okay, so it's very important. So let's talk about this business of whether astrocytes are involved in neuronal communication. So this isn't in the slides I released earlier. I was thinking about it this morning. And again, I was looking up the latest, so here it is. So it turns out that recently it's been really sort of better established that astrocytes can sense and respond to nearby neuronal activity. And they do possess receptor proteins for neurotransmitters. So these guys here can sense, to some extent, they have receptor proteins that can sense the presence of neurotransmitters. And when the neurotransmitter binds to those receptor proteins, running out of two screens. Oh. Perfect. This is going to work. Point where I discovered something was there. Oh, good. This is not the point where I discovered that some evil person left a perfectly respectable battery. Missed. All right, so put a perfectly respectable battery on that thing. I hope this one lasts. Let me know if it doesn't. All right. So what happens when the neurotransmitter binds to these receptor proteins is maybe the membrane potential of the astrocyte changes, or maybe its chemical messengers are released to change its metabolic properties, to make it move potassium, for instance, more readily across the membrane, or to increase its uptake of neurotransmitters and things. Okay, so it has receptors. I don't know this reference here. You can look it up probably from, but this has just got left on when I cut out the phrase. They also release neurotransmitters, particularly extracellular ATP, enabling neuron-glia-neuron crosstalk. So the neurons can release neurotransmitters onto the glia, and then the glia can respond by releasing some kinds of rather sort of primitive neurotransmitters back, like ATP, back onto the neurons. Right? And secondly, as you can see from this diagram, there's a really exquisitely sort of complicated, I wouldn't say it was refined, but complicated organization of glial cell processes and neuronal dendrites and axons. And this sort of intimate interaction may allow astrocytes to locally regulate vascular blood flow and nutrient delivery based upon neuronal activity. So the idea is that if these guys are very active, if neurotransmitters are being released on them maybe, the glial cells will detect those neurotransmitters and release factors that perhaps alter blood flow through that area or alter the level of ions in the extracellular space or change the sort of sculpting of neurotransmitter levels around the neurons, okay, that they perform naturally. And that this actually might be involved in information processing. Okay. If you want to learn more about this, there's a link here which is very faint here because I couldn't find the right font or couldn't figure out actually how to bloody change it. Anyways, there you can, when you download the slides, you can get it and click on it, and it's a review. And I haven't read it, I must say, but it's definitely a very interesting review, and I'll read it as soon as I get back. If you've got questions, let me know. Okay, so macroglial cells, there's others. Oligodendrocytes are responsible for myelination in the central nervous system. So they wrap around axons in the central nervous system and deposit those beautiful myelin sheaths. Okay, actually form those beautiful myelin sheaths. And then a separate class of glial cells called Schwann cells do the same thing in the peripheral nervous system. So glial cells make up the myelin. The actual cell membrane itself makes up the myelin that surrounds those axons that we saw. Here's the myelinated axons here surrounded by myelin. So these wrappings of membrane actually come from the membrane of nearby glial cells, either oligodendrocytes or Schwann cells. And then lastly, glial stem cells. As I mentioned, these can proliferate and differentiate into new glial cells, and as I said, in a very few places in the brain, into new neurons. And then there are separate glial stem cells, separate class, that makes new oligodendrocytes as well in the central nervous system. So people are just beginning to learn about these kinds of glial stem cells coming into focus. All right, so the neuroscience, yes.
Oh, sorry.
Sorry?
So earlier when you were talking about the myelin sheath, so
Yeah.
in the PNS, it's actually cells that create the myelin sheath. Like, so the sheath itself is like basically a bunch of cells.
The sheath are wrappings of the cell membrane of a single glial cell, actually. So each stretch of myelin is created by a glial cell, winding itself membrane again and again and again around a neuron.
So that happens in the PNS and the CNS?
Yes.
But
The but they're different names, different kinds of cells. One are Schwann cells and one are oligodendrocytes. And I don't know the evolutionary or ontological link.
Are there differences between the two, or is it just?
They look very different. Yeah, I mean they're not the same cell by two different names. But I don't know if you could ask me that on a discussion board, and I could look up how they differently differentiate, where they come from. Okay, so because of the structural diversity of neurons, any one part of the nervous system contains a bewildering array of anatomically distinct neuronal types, and also perhaps glial types as well. All right? Monopolar cells, bipolar cells, and so on. However, parts of the nervous system are often, so this is a mess, right, trying to figure this out, huge mess. But it turns out that parts of the nervous system, particularly in invertebrates and in more complex animals, parts of the nervous system are made up of layers. And each layer will contain a distinct, fairly limited population of specific neuronal types. Yeah. So you will have a layer in your retina, for instance, that consists largely of bipolar cells and one or two other cell types. You'll have a layer elsewhere, have other layers in your brain that consists of multipolar cells with a particular sort of kind of triangular cell body, a pyramidal cell body. The idea is that each of these layers contains a limited subset of neuronal types which interact together to perform a certain task or function in information processing. All right, so you have a part of the brain, and it's got these layers, and within the layers are particular kinds of cells, each layer. All right, so here's some different kinds of cells. Okay. And this layer, for instance, might be performing one little part of the overall calculation that's needed to process the information that's flowing through. And then the output of this layer would be sent through axons to this layer here, where the cells would perform another part of the calculation. And then here would be a third connection here to this layer. And eventually, these cells would output to somewhere. Come in here. Okay, so it's a little production line, right? A little miniature production line. And all you have to really know about it, apart from the general concept right now, is that it sort of simplifies the anatomy of the nervous system a little bit, these layers. So you can see these layers using special cytological stains. The Nissl stain, for instance, labels endoplasmic reticulum in neuronal cell bodies. And so this is a part of the nervous system that's stained with this Nissl stain, this purple stain. It's soaked up by the endoplasmic reticulum in the cell bodies. And you can see that this little part of the nervous system consists of layers of cells. And the different layers of cells have different sort of densities of cells within them, with slightly different sort of sizes of cell bodies and things. And so you can see that the density of the cell bodies here, for instance, and of the endoplasmic reticulum in these types of neurons is very high. Whereas here, there are very few cell bodies. Here's another layer of cell bodies, right? But not to quite so high density as here. And so using this sort of very gross method, you can actually distinguish these layers one from another and begin to sort of construct the microanatomy of a small part of it. So the Nissl stain is really good for distinguishing tissues and layers within the nervous system, but doesn't stain individual types well, because it doesn't stain the axons and the dendrites. It just stains the cell bodies of the neurons. Here it is high power. You see, you can't see the axons and the dendrites, just the cell bodies and the nucleus, because there's no stain in the nucleus. So then comes the Golgi stain, and we've looked at that. And that enables you now to go in and perhaps randomly stain some of these cell types. and describe the different kinds of neurons that are there. It's the next level. As an alternative to the Golgi stain, you can stain an individual neuron with a fluorescent chemical dye. This is not GFP, it's just a chemical. And there are two ways that you can get this chemical to stain an entire single neuron. And the first is to microinject the stain into a neuron. So you penetrate the cell membrane of a neuron, you microinject the fluorescent dye into the neuron. And then this will stain the whole of the cell, because it will diffuse through the cytoplasm everywhere within the inside of the cell. And will enable you to measure to look at the structure of a single neuron in a particular layer of the brain. This is really useful if you've previously been recording electrically from the cell with a microelectrode, an intracellular microelectrode. Because you can record from the cell electrically, and then at the end of your recording, you can pump dye into the cell, basically push dye into the cell, fill up the cytoplasm. And then you know, when you look with a fluorescent microscope later, exactly which cell you were making your electrical recordings from, and what it looked like. So that's an enormously powerful technique for figuring out who is doing what to whom in the brain. Right? How does an individual specified type of neuron in a particular part of the brain respond to stimulation of some kind? You can also, if you don't want to, if you're not recording electrically from the cell, you can also place a little droplet of a lipid-soluble dye onto the cell. And that lipid-soluble dye will get transported throughout the cytoplasm. So here's some examples. This is a neuron here that's been injected with a fluorescent dye, and you can see the very beautiful staining of the entire dendritic network of this cell. Here's another cell here. This would be the nucleus of that cell. These are cells that have been labeled with the Golgi stain, but of course the Golgi stain can only be used on dead cells in fixed section. It's a very lengthy procedure, so it's not much good for looking at cells that you've just recorded from in a living slice of brain. You can also, once you've made your recording, if you fix the tissue, you can also label it with antibodies. And maybe a particular protein will be expressed in the cell that you've been recording from. And that will enable you to say, oh, we've recorded from the cell that expresses this particular protein, and not another type of cell. Okay, so you can do, and you can make a little bit of sense of the kinds of cells in the section if they express different proteins, and you can use antibodies to distinguish one from another. All right, that's another general technique. Antibody labeling of proteins expressed only by a specific neuronal type. The other trick you can use to figure out what how this little network of cells is constructed is to actually place some fluorescent dye or some radioactive tracer onto the cell body of the neuron that is making contact with these cells, synaptic contact. So this would be the cell body of a neuron here, and if you infuse some radioactive tracer or fluorescent dye somehow into this cell, it may be carried through this synapse, along the axon here, it will be carried along the axon, and may actually cross the synapse into the next cell. And if you're really lucky, it'll be carried along the axon of that cell, and then cross the synapse to the cell after. And so you can actually use radio-labeled amino acids or fluorescent dyes to trace pathways through the brain. These sort of daisy chains of neuronal connections. Because when you inject material into one cell, if it's a particular kind of simple amino acid, radioactively labeled, or it's a particular fluorescent dye, it may cross the synapses between the cells. Okay, so that's another sort of neuroanatomical technique. And so this is a part of the brain that was that actually processes information from the eyes. So axons that come from your two eyes contact neurons in this part of the brain. It's called the lateral geniculate nucleus, right? And it's in your brain, and axons from the eyes contact it. And so this is part of the initial processing of visual information. And this part of the brain receives information, receives axons from both of your eyes. And so what's happened here is a radioactive tracer has been injected into the eye, one eye of an animal. A radioactive amino acid has been injected into one eye. It's taken up by the neurons of that eye. It's carried along their axons into this part of the brain. And then after a few days of this, you sacrifice the animal and slice that part of the brain up to create this section of the brain. And I think it's the dark areas here are neurons in this section in this part of the brain that were contacted synaptically by neurons from one eye. So the neurons in these parts, this part of the section, have the radioactive tracer inside them because they were contacted by neurons from this eye, say. Whereas these neurons here were contacted by neurons from the other eye. And so they're not labeled in the same way. And you can see, again, as in this diagram here, that the eye basically, each eye basically contacts 1, 2, 3, 4 layers within this little part of the brain. And the other eye contacts another set of layers. Right, so again, it's layering of information processing from one eye to the other. So that's radioactive labeling. What time do we have? Great, good, thanks. Yeah.
What was the difference between orthograde and retrograde on the previous slide?
Orthograde, so this will be an example of orthograde labeling, where you inject into the eye and it's carried into the brain. In other words, it's the radioactive tracer or the fluorescent dye is carried from along the axons from the cell body to the nerve terminals. Right? Retrograde is in the other direction. And so if you were to label these cells or inject radioactive tracer into these cells and they took it up, you might find that it's actually transported back through the synapse and winds up in one of the eyes, but not the other. And that would be retrograde. Yeah, orthograde. Also means usual method, retro means backwards. Okay, so last piece of, we talked about GFP. The last sort of new anatomical technique that I want to talk about, just mention, is to determine whether a neuron specifically expresses a protein. Not necessarily localizing where the protein is inside the brain, but is it simply inside a cell? But is it simply expressed by a cell, period? And for that you have to to engineer an enzyme, the code for an enzyme that produces a colored product of some kind into the neuron that expresses the protein that you're interested in. I think somebody mentioned this technique here. And when the protein is produced inside that cell, the enzyme is also activated, is also produced, and that enzyme can then be made to produce a colored product if you give it the right substrate. All right, so this is sort of an alternative to the GFP. It doesn't tell you where a protein is localized within a cell, but it can tell you whether a cell is producing a particular kind of protein. So all of these different anatomical and cytological and histochemical techniques have been used to try and figure out what the anatomy is of parts of the brain. And we'll see examples of their use many times as we continue with the course. Okay, so if you don't get the methods now, you will in a little while as we talk about the use. Okay, for the last part of this lecture, last 5 to 10 minutes, I want to talk about neurons as functional units. So electrical information generally, as we've seen, flows from the dendrites to the axon terminals, and then through synapses to other neurons. And electrical information flowing along neurons can take the form of all or nothing action potentials. That is, a spike in electrical activity, so that the membrane potential, what's a good membrane potential, resting membrane potential for a cell? Minus 60? Is that right? Minus 70? You prefer 70? All right. I take no responsibility for slides that have anything between 60 and 80. Right? And then, if the membrane potential rises above a threshold, an action potential is generated. Right? An action potential rises to plus 40, plus 50 or plus 60 millivolts, right? Somewhere in that range. Okay, so this is 0. Okay, all right? And it lasts for how long? Is it time? Time always confuses. How long does it last? Last a second? A millisecond, yeah, that's fine. Some last longer than that, especially in muscle cells. Could be up to 100 milliseconds. But mostly in neurons, it's a few more, a couple of milliseconds. All right. We'll talk about that. It's called an all or none event because once the membrane potential exceeds threshold, the action potential rises all the way to plus 50 millivolts. And it doesn't matter how it got to threshold, right? It's going to produce the same amplitude of action potential in a particular neuron. One neuron membrane potential, action potential may go to plus 50, another one to plus 40, another one to plus 60. But within a particular neuron, the dogma is that it's the same every time. So, information can only be carried through the number of action potentials that occur per second, the frequency of action potential. Since each action potential is a unitary all or nothing event, there's no information in the action potential itself. It's either there or it's not. It's a binary. Right? The only question is how many times does it happen per second? What's the bit rate? Right? The other kind of potential is actually the potential difference that raises the membrane to threshold in most cells. And this is usually a much smaller change in membrane potential that is graded in amplitude depending on the stimulus. We'll talk about what the stimulus means in a moment. Okay, and if the graded potential exceeds the threshold, about minus 60 or minus 50, usually minus 50 millivolts, then an action potential is generated in most neurons. Right? But there are neurons that have no action potentials. And if you inhibit the action potential by applying a toxin, for instance, you will see the graded potential still rising. If there's no action potential with stimulus intensity. Alright? And if you maintain the stimulus for a long time, the graded potential will often be maintained for a long time as well. Yeah. So those are called graded potentials. Now, action potentials carry information over long distances. That is, usually a millimeter or a fraction of a millimeter or more. Graded potentials carry information over very short distances, usually at the most a few hundred micrometers. So graded potentials are very important for carrying the information within the dendritic network of a cell. Along a dendrite, or from one dendrite to the cell body, and then back out to another dendrite. Whereas action potentials carry information long distances along axons. Okay, why that is, we can talk about later. But I just want you to get that clear right now. And the information in graded potentials is carried according to the height or amplitude of the graded potential, as well as its duration. The amplitude and duration. So bigger stimuli produce larger graded potential. So let's do some comparisons now. Yes?
Can you quantify the graded potential as not modulating threshold?
You mean, so what I said is if you limit the action potential, you will see the graded potential go above threshold. Yes? If you block the voltage-gated sodium channels, for instance.
So normally graded potential is still 50.
No. To get, how do you think you get from here to threshold? With a graded potential, all right? So they're tremendously important in triggering action potentials, right? And they're tremendously important for carrying information about how much neurotransmitter is arriving on a dendritic network and where. Yeah? But in a long distance perspective, all that they do normally is trigger the action potential. All right? Go ahead. Yeah. Everybody else get that exchange? So let's do a comparison with computers. I don't have a watch up there. I have a couple of minutes.
You have 9 minutes.
I have 9 minutes. No, it's 1:35 now. That's 5 minutes. No, that's pretty much gone. Right? Let me just get through this slide. Okay, so let's make a comparison with computers here, right? And let's go to our laptop open, right? How fast is your laptop? What's the speed of your laptop? Anybody? 3 gigahertz, right? That's pretty good. Okay, 3 gigahertz. All right, so that means 3,000 million bits of information are transmitted, or actual operations are processed by the processor in that computer every second. All right? But a single neuron is limited to about 1,000 bits per second of information capacity. Because at most, we can generate 1,000 action potentials per second. Each action potential lasts 1,000th of a second. All right? But we know that our brains are smarter than computers. We know that we can process visual information faster than certainly a small laptop can do in real time. And the way we do that is because we have millions of neurons, all of which are looking at the visual world at the same time. And so we are massively parallelly processing the information that comes into our brain. So instead of having one processor in a computer, or almost a couple or 4, that processes information at gigahertz rates, we have a million processors in terms of million neurons or million neuron pathways in those layers that process information each at 1,000 hertz rates. All right, so that is the essential difference between our ability to process information and a computer's ability to process information. Okay, so that's all I want to say on that subject.